Testing, 1, 2, 3, testing. So good morning, everybody. I'm going to try our first journal club today. My name is JC, and this is JC on a bike. I'm going to bike into the University of Pittsburgh out here from Shaler. It's about a 24-minute commute. So that gives me about just the right amount of time to do a couple takes of introducing a paper to you that maybe then I'll try to describe a little bit more about on the way home. So the paper that we're going to discuss today is a paper by Gilad, Silverberg, and Henry Markram. Henry Markram is kind of infamous for being the guy who got $1 billion from the EU for I think it was the Human Brain Project, but he's also the blue brain guy, the guy who tried to simulate a cortical column on a supercomputer. And he's kind of controversial because some people think he's just a lot of big talk. But, you know, to a certain extent, everybody that gets grant money is a little bit big talk, so I'm not really judging him here. This is a great paper, and it's a great discovery. In the paper, what I like most about it is that it's full of really, really beautiful data. Now, there are some caveats to this beautiful data. And some of those caveats are obvious, and some of them are not, but I think the first and foremost one that everybody's going to hammer on is the fact that this paper is done in juvenile mice, 14 to 16 days old. And from an electrophysiologist perspective, that means that this paper is done in very healthy, very intact slices. And if you want to argue that baby slices are baby bullshit, then that's different. But good morning. So the paper is really simple in the sense that it makes observations, and I think that's where the best electrophysiology papers are rooted in observation. I think now we tend to think of, a lot of times we use electrophysiology as sort of a check. Turkey over there. As sort of a check for something else. You know, how does this gene function? Is this expression really there? It's become a tool that we use to just prove shit that we already know in our head we want to prove, and that to me is a lot of times a very frustrating part about being an electrophysiologist. I like it a lot better when people say, hey, I got this new cell type. I'm interested in having you help me find out what it does, how it works, what's unique about it. And as luck would have it, I'm just not a patch clamp physiologist during that time. It's harder to make beautiful discoveries in vitro now, because a lot of the usefulness of patch clamp physiology, some might even argue most of the usefulness of in vitro patch clamp physiology of any sort, is that the resolution is the single neuron. You can see synaptic currents, record synaptic events, you have great control over pharmacology, And if you try to do your experiments with some thought, you can avoid the problem of not really being able to voltage clamp the entire neuron. Now, for the lay people out there, you don't really need to know what that is right now, but if you keep tuning into my channel, I think eventually you'll figure it out, or we'll learn it again together. But in patch clamp physiology, you're able to measure very distinct, minute details about how ions move in and out of neurons and how neurons signal to one another. And so, you know, if you ask yourself, what the hell is a patch clamp physiologist? It's somebody who studies how neurons function, how cells function. In this case, a neurobiologist who would apply patch clamp physiology to neurons. And, you know, we've come a long way into understanding how neurons work. And now the sort of drive in neuroscience is to understand how neurons work together. As a patch clamp physiologist, it becomes more challenging to stay on the cutting edge of science and not fall back into this, you know, glorified technician position where, yeah, you can do really cool experiments. I have this really cool molecule. Maybe you can do some cool experiments for my molecule and we can make a paper. Those are fun, but it just doesn't feel like genuine discovery. And this paper by Gilad and Henry really is genuine discovery. So if we start with figure 1, figure 1 is a basic observation, and I think that's what makes this paper foundationally stand out to me as something that is to be modeled or emulated. Because here they start out with an observation that on its face essentially begs the reader to make her own hypotheses about what this observation could be. Here's 2 pyramidal cells. We record them simultaneously, and when we stimulate one pyramidal cell with a 70 hertz 15 action potential train, we see emerging in the other layer 5 pyramidal cell a hyperpolarizing signal, which in the exclusive dichotomy of synaptic physiology, that should be GABA. It should be inhibition. It should be a signal trying to turn the recipient neuron off or make it less excitable. It's the opposite of excitation. It's not turning him on, it's turning him off. And so they show you data from several recordings. And I know Gilad, he was making 7 patch recordings, so 7 pyramidal cells at the same time. And then stimulating them with trains. Come on, dude. And then when those trains resulted in inhibition over there in the postsynaptic cell, yeah, get some. That you then just average it, right? You find the connection that has it, and then you average those, so you can get a good idea of sort of size and latency, that kind of thing. And so you see that it requires a lot of action potentials. It won't happen on one. And so as synaptic physiologists, and Gilad and Henry already, you know, had thought with other papers, had already sort of explored the idea that the way a synapse releases during a train could essentially function as a high pass or a low pass filter for neurons talking to one another. And so they already see that here. We don't need turn signals in Pennsylvania. So keep in mind, that's just the observation, right? But the observation is clean. And it's just presented, you know? You don't need any analysis or error bars from control to convince you that this is a circuit, because it's a freaking circuit. And it's a repeatable observation. So I think figure 2 now shows another doublet, so 2 pyramidal cells. But now when they look at these doublets and they look at how they're connected, and they look at slow trains, so figure 1 had this fast 70 hertz train. Now they're looking at 20 hertz trains and 10 hertz trains of action potentials. And they see that these trains can sometimes evoke what appears to be 2 loops, or 2 inhibitory signals. One that seems to have a fast kinetics and be evoked on the first action potential, and then the other appears to be a slower one whose kinetics resemble the loop, the disynaptic loop that was observed in figure 1. So this becomes cool, because then what they do is they pair a pyramidal cell with local interneurons, and in this case they do bitufted or multipolar, and these happen to be the same 2 interneurons that Reyes et al. Out of Bert Sackman's lab showed to receive different facilitating or depressing synapses from pyramidal cells already, I don't know how many years before this paper. So this observation is very confirmatory of that paper, and is now going to put those observations that Reyes made, along with Nail Bernyshev and Andrei Razov, they're going to put those observations in the context of a microcircuit. So now we're going to see the consequences of having facilitating or depressing short-term synapses from a pyramidal cell to an interneuron. And so that's pretty cool, because when they look in figure 2 and they look at pairs between interneurons and pyramidal cells where the pyramidal cell is synapsing on the interneuron, then you can see the expected synaptic dynamics are there already. You shithead. Come on, guys. We gotta drive here. What are you doing using your brakes right now for? There's no car in front of him. Anyway. So when they record from a bitufted cell that has an excitatory synapse originating in a layer 5 pyramid, they see this excitatory synapse is facilitating a train. And when they look at a fast spiking cell, multipolar interneuron, they find the depressing synapse. So the first action potential is the one that triggers the interneuron to fire. So although this is confirmatory evidence of this differential synaptic contacts from one pyramidal cell onto different postsynaptic interneuron subtypes, this paper is about to put it into the context of a microcircuit, because we're trying to explain the observation in figure 1, right? These subsequent experiments are to figure out what's happening in figure 1. And again, it's hard to find electrophysiology papers that present a discovery or a finding in this way. And I think it's a great paper in Neuron, but it should have been higher. So, and the reason why it should have been higher is because they do it so thoroughly. So, wow, the sumac is already changing along the river a little bit. Yeah, so let me think about what figure 3 is again. I don't remember anymore. Yeah, so figure 3 then takes this loop, and when they find the loop, and that's the trick, right? You patch 7 cells, a couple of the pyramidal cells have a interneuron loop between them, and then you work on that pair and ignore the rest of the 7. And in a young slice, that's a pretty doable experiment. You can regularly get 6 or 7 cells if you're talented, and Gilad is talented. So you take a look at one of these slower loops, and you just do the frequency dependence of it. So how many action potentials? We're going to get 15 all the time, but we're going to put 15 action potentials in different spacings. So essentially we're going to change the frequency that this filter is going to allow through, and you're going to see that as the frequency of the incoming train increases, so the action potentials are closer together, then what happens is that you're more reliably able to activate the interneuron loop, and even better, you get a higher amplitude, because likely you're getting more action potentials per depolarization. And that comes again because you're putting them closer together. If you remember the excitatory synapse that they show in figure 2B, then you can see why that works, because in B you see this synapse, in 2B you see this synapse where it's facilitating. So the more action potentials that you can put closer together, the more likely you are to get a spike. And if you get that close enough together, you're going to get a whole train of spikes. Green arrow or green light, could be, depending. Green arrow. Police officer, I don't care. And so now we go on to figure 4. Well, first let's say figure 3 again. In discussing figure 3, it's important to see that this is also just it's an experiment that you can do in succession with the other observations that they're making here. In any given cell where you have 2 pyramids on line and they have a loop between them, you can do a lot of the experiments in figure 1, and the experiments in figure 2 are figure 2A, and the experiments in figure 3. But you have to have an idea of what to do, and you need to be a good enough electrophysiologist to get a stable enough recording to make those measurements over time. None of these measurements are trivial. If you want to make a paired recording like this and have mean responses that look as clean as this, I think you better be prepared to do at least 20 sweeps of every trace that you want to show. So in figure 3, where he demonstrates the frequency dependence, each one of those traces is an average of 20, probably, maybe 30 sweeps. That's a long, stable recording then, if you think he did 4 frequencies, or he washed out pharmacology, as he does in the next figure. So, in figure 5, figure 4, you have this. In figure 4, what you're really looking at is simple pharmacology, of course, and 2 different demonstrations of how you can differentiate a putative GABA signal from what should be an AMPA-NMDA excitatory signal. Now, the first way to do it is just use pharmacology. Bicuculin is a dirty blocker. It blocks a little bit more than GABA channels. It blocks some potassium channels too, but if you want to demonstrate what a hyperpolarizing current is in this scenario, where you need to spike a cell to see it, and you can kind of make the jump that this is a synaptic signal, then you only really have GABA or glutamate to block. GABA, the inhibitory transmitter. And so, in that sense, blocking with bicuculin is fine here. Washing bicuculin is a challenge. You can see that the wash never comes back to the full control level. And that's because bicuculin is pretty sticky, and the fact of the matter that he can wash it out at all is, in my mind, already demonstrates the stability of his recordings and the validity of his observation. So, the other way to show that it's GABA would be the more challenging way, perhaps, and it also you gain some information with this technique because you can demonstrate whether or not the signal that you're seeing in this loop, this putative GABA loop, you can demonstrate whether this signal is sorry for the swearing. We don't need turn signals in Pennsylvania. The other way to do the GABA thing would be to patch dendrites. So you have a pyramidal cell, and lucky enough, pyramidal cells are nicely organized morphologically. They have a soma, and some dendrites around the soma, and then they have an apical dendrite, which has its own special electrical properties and tends to be thought of as a different compartment. So, happily enough, this morphology comes in handy in the sense that we have long thought that the apical dendrite is a sort of separate computational compartment of a pyramidal neuron. And so, patching the apical dendrite is really patching another computational unit in a way, or at least a subunit of the pyramidal cell. And so, if they're lucky and their hypothesis is correct, because of the reversal potential they see, they can at least see whether this inhibition is coming in on the basal dendrites or the apical dendrite. And if it's coming on the apical dendrite, then their hypothesis is that the signal that they measure from the dendrite versus the signal they measure from the soma, they should see a stronger inhibitory and faster inhibitory signal the closer they are to where the synapse occurs. And so if it occurs on the apical dendrite and they patch the apical dendrite, that should be a better signal than the somatic electrode. And so that's what the second half of figure 4 is showing. That's what figure 5 is showing, I believe. Yeah, so I think figure 4 is the one that actually shows reversal potential and does bicuculin. And so then, thank you. So then, and then you're not going to go anywhere with your hillbilly truck blowing smoke in my face. Then you're not going to drive at all, right? You're going to stay over here to the right. Yeah. Go ahead. So, figure 5 then demonstrates that this inhibition is likely to come in on the apical dendrite. And so now the only two things left to do really are the triplet and the morphology. And so these experiments are still to this day the sort of gold standard for connectivity in the cortex or anywhere else. you patch all the cells that are connected, right? That would be the way to show it. And so that's what they do. And that is not trivial. They are going to patch the pyramidal cell, they're going to patch the bipolar interneuron that he's attached to, and they're going to patch the pyramidal cell that gets inhibited by the bipolar cell. And in this case, they actually show you a group of pyramids, two pyramids that can both are inhibited by the same interneuron, and one of them can activate it. And then in figure 7, they show you the morphology, and I think this again is just a lost art, right? I mean, only a few labs like Bernardo Rudi's lab in America and in New York, and a few really awesome labs in Japan are really able to nail down a morphology-based comparison of neurons. It's a challenging thing to fill a neuron. I've been patch clamping for more than a few years, and I find it pretty difficult still to get consistent fills. So that's the paper. Maybe I don't need my afternoon ride, I don't know. But I can give you a summary of it again, just to see how well I know it now. So this paper starts with figure 1, which is just an observation that if you stimulate one pyramidal cell in layer 5 with a high frequency train, you can observe what appears to be an inhibitory signal in other pyramidal cells. And that inhibitory signal in figure 2 is shown to have a frequency dependence. And so in figure 2 they show a double loop where a slower train of action potentials can bring about a very fast loop and then a loop on the same time frame as the loop shown in figure 1. And those two loops, they hypothesize might be from different types of interneurons because they know already of this paper by Reyes et al. That shows that these interneurons can receive different short-term synaptic plasticity between pyramidal cells and themselves. I don't know how I'm saying this, blah, blah, blah. But synapses from pyramidal cells can be different from the same pyramidal cell depending on the target cell. And so they are hypothesizing that this is indeed what underlies these two loops, the fast loop and the slow loop shown in figure 2. But the rest of the paper is focusing on developing and identifying the mechanism behind the slower loop. So figure 3 shows the frequency dependence and the number of action potentials necessary. So A and B is different frequencies of trains. C and D is action potentials in a train. And this is again demonstrating the sort of high pass or low pass, the sort of filtering characteristics of this connection. What does it send through and what makes it and what doesn't. It tends to be trains and it tends to be higher frequencies. So a high pass filter. Figure 4 is showing that it's GABA, blocking bicuculin, and also reversal potential as expected around minus 70, 75. It actually is a little lower than that, despite being in whole cell. That's why they also make the jump to the next figure where they make dendritic patch, because the lowered reversal potential does suggest that the signal's not coming into the soma, otherwise the reversal potential should be around the reversal potential for GABA of the pipette solution. So then they make dendritic patch recordings, and they show that they can see a bigger signal in the apical dendrite, and it's faster, and that suggests that the synapses between the bipolar cell and the pyramidal cell, the inhibitory synapses from the bipolar cell, are likely to be on the apical dendrite. And then the last two figures are just the triplet, which is a bombastic figure, where they record from the pyramidal cell, the interneuron, and the postsynaptic pyramidal cell that receives the inhibition. They can demonstrate all the things they've demonstrated in the first four figures with the triplet, and the bonus that they get from the triplet is that they can also fill the cells. And when they do that, they're able to count contacts and see that the contacts are biased toward the end of the apical dendrite, blah, blah, blah. It's super cool. Now you try and go in the literature and find another paper that's so thorough and that so nicely leads you by the hand to understand what their observation is. I don't think you'll find one. At least not one that beats this paper. It's a fabulous paper. And forever, Gilly and Henry should be proud of it. What do you think about them apples? Hey, so thanks for joining me on my ride. That was the first JC on a bike, Journal Club on a bike. We covered the paper in Neuron from Gilad, Silverberg, and Henry Markram, disynaptic inhibition between cortical pyramidal cells. We now know that this is likely a Martinati cell and likely a somatostatin positive interneuron. The paper is chosen specifically because they start with an observation. And I think there are so many papers nowadays that don't start with an observation, they start with an assumption based on other people's work, and they always start in the middle of the story. This is one of the few papers out there, and one of the best examples of a paper out there, which takes an observation as simple as stimulating one pyramidal cell and seeing inhibition in the other, and then using painstaking electrophysiological techniques to reconstruct this circuit and to convince you of the mechanistic underpinnings of how it works, what the synapses are like, and why the short-term dynamics of the synapse are what determine how this loop shows up. And incidentally, in this paper, they are also showing the fast spiking cell loop, although it's not something that they necessarily emphasize in this paper. It's already clear to see in figure 2, where they show that a fast spiking cell can facilitate a short latency low-pass filter and the Martinati connection, because of its facilitating synapse, driving it from the pyramidal cell, functions as more of a high-pass filter. This is a very interesting finding. It's something that everyone should be incorporating into their understanding of the cortex. Even though this is done in baby slices, you still need to incorporate it, because the likelihood that these interneurons are inhibitory connections are actually important, it's definitely true. I can tell you right now, as we go through the brain over the next couple years with me, if this continues that long, you're going to see that interneurons are actually the boss and that the excitatory cells are just slaves. Anyway, thanks for joining me. I really enjoyed doing this journal club. I'm looking forward to doing the next one. If you liked it, please like and subscribe to JC on a Bike, and I'll see you next week.